Bile is a greenish liquid that's made by the liver and is stored in the gallbladder. Bile's a bit like an alkaline soup, and its ingredients include a variety of organic molecules. Bile does a number of things, including helping with digestion, absorption of fats, and excretion of various molecules. Normally, lipids are insoluble in water, so that's why bile is needed to help emulsify and solubilize them. The organic composition of bile is mainly made up of bile salts and phospholipids, with cholesterol and bile pigments, called bilirubin, making up only a small percentage. Bile is first manufactured in the liver by cells called hepatocytes. Hepatocytes use an enzyme called 7-alpha hydroxylase to convert cholesterol into two primary bile acids, called cholic acid and chenodeoxycholic acid. In the intestines, some of these primary bile acids get dehydroxylated, giving rise to secondary bile acids, deoxycholic acid and lithocholic acid. The liver can conjugate or attach the amino acids glycine or taurine to all four of these bile acids, and this ultimately gives rise to 8 different forms of bile salts. And it's these bile salts that are the main components of bile. So, bile is made in the liver and flows into the intestines. The journey starts when bile flows into the left and right hepatic ducts, which eventually merge to form the common hepatic duct. The common hepatic duct then leads to the cystic duct, which brings the bile to the gallbladder. The gallbladder is a small pear-shaped, hollow organ located beneath the liver, and this is where bile is stored and becomes more concentrated. Approximately 30 minutes after consuming a meal, the food is broken down into a slurry called a chyme, and the chyme begins to enter the first part of the small intestine, the duodenum. When that happens, eye cells, which are in the mucosal lining of the intestine, secrete a hormone called cholecystokinin, or CCK, into the bloodstream. Cholecystokinin travels through the blood and reaches the gallbladder, causing it to contract really strongly. These strong contractions cause the stored bile to get squirted out of the gallbladder and into the common bile duct. The bile ducts are lined by cells called cholangiocytes, which secrete bicarbonate-rich fluid into the biliary tree. Cholecystokinin also causes the relaxation of the sphincter of Oddi, the muscular valve between the common bile duct and the duodenum. This allows the bile and the bicarbonate-rich fluid to flow through the sphincter of Oddi and into the duodenum. Bile salts are amphipathic, meaning that the molecules have parts that are hydrophilic or water-loving and other parts that are hydrophobic or water-fearing. This unique structure allows bile salts to self-assemble into mixed micelles when they enter the intestines. Micelles have a hydrophilic exterior and a hydrophobic interior, and little droplets of fat from the diet can then settle within this hydrophobic interior. This is how bile salts emulsify or break down large fat globules into small emulsion droplets. If you compare a large fat globule to many small ones, the biggest difference is surface area. In other words, by emulsifying the fat, micelles help generate a much greater surface area for the enzyme pancreatic lipase to digest lipids into fatty acids and monoglycerides. Micelles move lipids to the brush border of the intestinal cells where they diffuse out and are absorbed through the intestinal villi. Without the help of bile salts, lipids wouldn't get digested and absorbed, and they'd get excreted in the feces, causing steatorrhea, an oily type of stool. In addition to lipid digestion and absorption, bile acids also help with bilirubin excretion. Bilirubin is the pigment that gives bile its color, and it's actually a byproduct of hemoglobin metabolism. As red blood cells near the end of their lifespan, which is about 120 days, they're destroyed by immune cells like macrophages. macrophages break down hemoglobin, which are iron-rich proteins inside blood cells that bind oxygen into heme and iron. Next, an enzyme called heme oxidase converts heme to biliverdin, which is green. Then, the enzyme biliverdin reductase converts biliverdin into bilirubin, which is yellow. Finally, the bilirubin leaves the macrophage and enters the blood. The bilirubin leaving the macrophage is not water-soluble and is therefore referred to as indirect or unconjugated bilirubin. Albumin in the blood binds to the unconjugated bilirubin in order to give it a lift over to the liver, where it's taken up by hepatocytes. Within the hepatocytes, the enzyme uridine glucuronil transferase conjugates the unconjugated bilirubin to glucuronic acid, turning it into conjugated bilirubin. And the conjugation of bilirubin allows it to become more soluble. As a result, conjugated bilirubin can be secreted into bile, whereas unconjugated bilirubin is typically not found in bile. So ultimately, bile containing the unconjugated bilirubin flows into the intestines. In the intestines, certain bacteria hydrolyze and reduce conjugated bilirubin into a colorless compound called urobilinogen, which can enter various pathways. Most of the urobilinogen, also called stercobilinogen when it's in the intestines, becomes oxidized by gut bacteria to form stercobilin. Stercobilin is what makes feces brown in color. Now, there's a system by which both bilirubin and bile acids get recycled, called enterohepatic circulation. A small amount of urobilinogen that's not excreted in the feces gets reabsorbed from the gut into the bloodstream. Some of that urobilinogen travels through the blood to the kidneys, where it's converted to urobilin, which is very water-soluble and therefore excreted into the urine and gives urine its yellow color. The rest goes right back to the liver to get sent out into the intestines once again. Now, unlike bilirubin, where only a little bit is recycled through enterohepatic circulation, it turns out that roughly 95% of the bile acids get recycled that way, and only about 5% are lost in feces. In fact, there are sodium bile salt cotransporters within the intestine which specifically bring bile salts back into the enterocytes. These bile acids get into the blood and ultimately back into the liver so that they can get added back into the bile once again. Alright, as a quick recap. Bile is formed by the liver, stored in the gallbladder, and released into the duodenum. Bile salts are able to emulsify large lipid globules into smaller droplets to create a greater surface area upon which pancreatic lipase is able to work. Aside from bile salts, another component of bile is a pigment known as bilirubin, which is a byproduct of red blood cell degradation. Both bile salts and bilirubin can be recycled via enterohepatic circulation, where they're transported from the intestines back to the liver.
Is a long, skinny gland the length of a dollar bill and is found in the upper abdomen, or the epigastric region, behind the stomach. It has endocrine functions, meaning it secretes hormones into the blood that eventually act upon their target tissues. For example, alpha and beta cells in the pancreas make hormones like insulin and glucagon that are secreted into the bloodstream to help regulate blood sugar levels. However, approximately 90% of the pancreas is dedicated to its exocrine functions. The exocrine pancreas secretes enzymes and fluids that help neutralize and digest food within the intestines. The exocrine pancreas can be divided into lobules, each of which contain lots of functional units called acini. An acinus is a cluster of acinar cells that all work together to make digestive enzymes. In fact, the word acinus means berry, which describes the berry-like appearance of these cell clusters. Each acinus secretes digestive enzymes which flow into small intercalated ducts that are lined by ductal cells. These ductal cells secrete bicarbonate in fluids that make up the liquid portion of pancreatic fluid, and ultimately help to neutralize the acidic stomach contents as they enter the intestines. The intercalated ducts merge together, forming an intralobular duct, which join with other intralobular ducts, and finally drain into the main pancreatic duct. The main pancreatic duct travels through the length of the pancreas and drains into the duodenum. Now let's look at the enzymes made by the acinar cells. The main enzymes include pancreatic amylase, which breaks down carbohydrates, trypsin and chymotrypsin, which break down proteins, and lipase, which breaks down lipids. Now, as it turns out, macromolecules like carbohydrates, proteins, and lipids are also found in the cells of the pancreas. Therefore, in order to protect the pancreas from destroying itself, the acinar cells manufacture inactive forms of the enzymes called proenzymes, or zymogens. These zymogens need to be activated by proteases, which cleave off a polypeptide chain, kind of like pulling the pin on a grenade. For additional security, the zymogens are kept away from sensitive organelles in storage vesicles called zymogen granules, and are packaged with protease inhibitors that prevent enzymes from doing damage if they become prematurely active. To digest a meal, these zymogens are released into the pancreatic duct and delivered to the small intestine where they're activated by the protease trypsin. Trypsin is a pancreatic digestive enzyme that's initially produced as the zymogen trypsinogen. Normally, trypsinogen isn't activated until it's cleaved by the protease enteropeptidase, which is found in the duodenum. Now, two enzymes that don't need activation by the protease trypsin are pancreatic lipase and amylase, both of which are secreted in their active forms. Now, the secretions of the ductal cells are filled with various ions, particularly sodium, potassium, chloride, and bicarbonate. The concentrations of sodium and potassium are about the same as the concentrations in the plasma, whereas the concentrations of chloride and bicarbonate depend on the pancreatic flow rate, which is the volume of secretions per minute. The chloride and bicarbonate concentration is regulated by transport channels on ductal cells. On the apical membrane, which is the side of the ductal cell facing the duct, there's a chloride bicarbonate exchanger. And on the basolateral side of the membrane, or the side facing the extracellular fluid, there are both sodium-potassium ATPases and sodium-hydrogen exchangers. Within the pancreatic ductal cell, the enzyme carbonic anhydrase combines carbon dioxide and water into carbonic acid. Carbonic acid then dissociates into bicarbonate and hydrogen. The hydrogen ion is pumped out of the ductal cell and into the blood, in exchange for a sodium ion via the sodium-hydrogen pump, and a bicarbonate ion is pumped into the pancreatic duct, becoming part of the pancreatic fluid, in exchange for a chloride ion. When food is being digested in the intestines, the concentration of bicarbonate in the pancreatic fluid has to be high to offset the acidity of the stomach acid. Consequently, the chloride concentration in the pancreatic fluid will be low, since the chloride ions are being exchanged for bicarbonate ions. So when the pancreas increases secretion, which increases the flow rate, the bicarbonate concentration increases and the chloride concentration decreases. Now, there are three phases of pancreatic fluid secretion: the cephalic, gastric, and intestinal phases. Let's say you're about to eat a nice, big slice of pizza. In the cephalic phase, the sight and smell of the pizza reaches the brain and causes it to send efferent signals through the vagus nerve to the pancreas. The vagus nerve secretes the neurotransmitter acetylcholine, which stimulates muscarinic receptors on acinar cells and causes them to secrete enzymatic fluid. Next is the gastric phase, which is when the chewed up pizza enters the stomach and makes it distend. Distension is sensed by mechanoreceptors in the stomach wall. This afferent information travels through the vagus nerve to the brain, which sends efferent signals back through the vagus nerve to the pancreas. The result is that the acinar cells produce slightly more fluid. Finally, there's the intestinal phase, which is responsible for over 80% of pancreatic secretions. When chyme, or partly digested food and stomach acid, enters the duodenum in the intestinal phase, proteins and lipids are broken down into various components like amino acids and fatty acids by pancreatic enzymes that get produced in the cephalic and gastric phases. These amino acids and fatty acids stimulate eye cells, which are in the lining of the duodenum and jejunum. Eye cells secrete the hormone cholecystokinin, or CCK, which binds to receptors on the acinar cells of the pancreas and strongly stimulates them to increase secretion of digestive enzymes. Chemoreceptors in the intestinal mucosa can also detect the presence of lipids, carbohydrates, and proteins, as well as decrease the pH in the lumen. These chemoreceptors send afferent information to the brain via the vagus nerve, and efferent information comes back to the pancreas through the vagus nerve to increase pancreatic secretion during the intestinal phase. Similar to the acinar cells, ductal cells also have cholecystokinin and acetylcholine receptors. In addition, ductal cells have receptors for a hormone called secretin, which is released during the intestinal phase. Partially digested food arriving from the stomach is acidic, and when it enters the duodenum, the rise in acidity signals S cells that line the duodenum to release secretin. Secretin acts on secretin receptors on the ductal cells to massively increase secretion of the aqueous portion of the pancreatic fluid, which is high in bicarbonate, and therefore helps to neutralize the acidic chyme. When food eventually reaches the ileum and colon, neuroendocrine cells secrete the hormone peptide YY, which helps inhibit pancreatic secretion. Although the exact mechanism here is not well understood. Alright, as a quick recap. Acinar cells of the pancreas produce enzymes that help digest food, and ductal cells produce aqueous secretions that help neutralize the acidic stomach contents. Acinar cells are mainly stimulated by cholecystokinin and the neurotransmitter acetylcholine, whereas ductal cells are mainly stimulated by secretin. Finally, the concentration of sodium and potassium in the pancreatic secretions is relatively constant, but the concentration of bicarbonate increases when there's a higher demand for acid neutralization.